Dr. Vasudev Bedekar, Dr. V.N. Bedekar, who has founded the current campus, as Anita Madam said, Nanavati, 13.5 acres of educational campus, housing institutes of higher learning. We have a polytechnic, we have management institute, arts and commerce college, science college, and then of course a law college, which is a postgraduate law college. After graduation, students can join for law. And we have advanced study center. All these higher learning institutes are housed in this campus, and there are auditoriums, a big auditorium, small auditoriums, a cafeteria, security building. So it's a self-sustained educational campus founded by Vidya Prasarak Mandal, and principally it is the idea of Dr. V.N. Bedekar sir. Around 60 years ago, there was no college in Thane, and that is the time he thought that our students should not go in the crowded trains. That time's crowded trains. Today the crowd is, I think, 100 times more. So why should students go? That time the nearest college was Ruya, and later on Somaya was founded. So why should they go up to Ruya? Or if they want to go Kalyan side, there was no college up to Ulhasnagar. So students had to travel right up to Ulhasnagar. So that is the vision, that we should have a college in Thane. And that is how our integrated arts, commerce, science college was established. And today we have arts and commerce college known as Joshi Bedekar College and science college, Bandodkar College, which are named after the donors. And of course the other institutes. And about 15,000 students coming in daily. This has been disrupted a bit due to the pandemic, but we are coming back to the original activities which were there 2 years ago. So once more, let me welcome you, madam, and we are all eager to listen to you. So now I hand over the mic to Parmar, Lakshmi Parmar. Well done, Lakshmi. Well done, Department of Chemistry. I would also request the Department of Chemistry, if possible, tomorrow, if we can meet for alumni interaction. We can invite an alumni. I'm trying somebody, and have a small interaction of alumni in the department. Whatever students can come and whoever staff can come around 2 o'clock in the afternoon. Yeah. So thank you so much, madam. Welcome. Namaskar.
Okay, sir. Thank you so much. Thank you
Please,
so much, sir. Thank you.
please carry on the program. I'll be here, and I want to listen to the new developments in drug discovery. So please, thank you, Anita madam, for such a nice program.
Okay. Thank you so much, sir.
Yeah, thank you.
In remember of late Dr. V.N. Bedekar, sir, this colloquium lecture has been arranged by Department of Chemistry. It is a well-known and primer educational trust in Thane, which was established in August 1935. It was the dream of founder, member late Dr. V.N. Bedekar, sir, to create an island of knowledge, Jnandipa, in Thane, and unconditional support from his colleagues and seedling growing into a tree, Balkrishna Nayak Bandodkar College of Science is one of the 17 institutions belonging to VPM. Our institute has great legacy of Gyan Tapasvi, Dr. V.N. Bedekar sir, continuing his legacy, who is the present leader, Dr. Vijay Bedekar sir, chairman of VPM. In 1969, Vidya Prasarak Mandal College started functioning with all three faculties, arts, commerce, and science. V.N. Bandodkar College of Science was separated from the Arts and Commerce College for administrative convention. V.N. Bandodkar College is the only single faculty science college affiliated to University of Mumbai. Our institution welcomes the rationale behind the ideologies of PhD with great passion. As we all are concerned about today's topic, impact of bioisosteres in drug discovery. Basically, bioisosteres are a chemical substitution or group with a similar physical or chemical properties, which produce broadly similar biological properties to another chemical compound. Taking the event further and know more about it, I, on behalf of everyone present here, would like to welcome our today's guest, Dr. Anita Abhyankar, ma'am. I request my friend Sneha Tiwari to give a brief introduction of ma'am. Over to you, Sneha.
Sure. Thank you, Lakshmi. It's my honor to introduce our today's guest, Dr. Arundhati Abhyankar, ma'am. Ma'am has completed her HSC and SSC from Mumbai board. Then she went for Bachelor in Pharmaceutical Science from Mumbai Educational Trust, Institute of Pharmacy, Mumbai, and Master of Pharmacy Medicinal Chemistry from ICT. Even PhD from ICT with credit course of Organic Reactionism and Stereochemistry. Ma'am has published her 13 papers, 89 citations, 8 poster presentation, and ma'am also has 2 provisional patents. In academic, ma'am has 4.5 years of experience as assistant professor in pharmaceutical chemistry at Bhanuben Nanavati College of Pharmacy. In industry, she have 1 year of experience. Most amazing thing to know is she had received Best Student of College Award in 2005 and JRD Tata Scholarship in 2004. Ma'am also received Sir Ratan Tata Trust Scholarship in 2004. Ma'am got Fellowship of UGC and DBT in year 2006 and 2009 respectively. In 2012 till 2015, she was a Fellowship of CSIR Senior Research. Of course, Ma'am Arundhati have many more achievements lining in the way, but these are the few. And it's really an honor to have such a great personality in between us today. Yeah, so it's a brief introduction of Ma'am. Now I would like to request Lakshmi to take over and continue.
Thank you, Sneha. It was really wonderful introduction of ma'am. Now I request Dr. Arundhati Abhyankar ma'am to take over and shower us with her beautiful words. Over to you, ma'am.
Thank you, Lakshmi. Thank you, Sneha. That was a very kind introduction. More importantly, I would like to thank Goswami Madam and Moses Sir for organizing this wonderful lecture and giving me an opportunity to present in front of you all. More importantly, it means a lot because this lecture series has been planned in memory of a great philanthropist, a visionary, and it's one of the rare opportunities for us to present for such a unique event. So thank you once again, Madam. And I also thank the management at my end for allowing me to present at such an event. So I'll just share my slides. If there is any issue, please let me know.
No, ma'am, you can.
Okay, I hope the slides are visible.
Yes, madam, it is very much visible.
Okay. Yeah, thank you, madam, for confirming. Also, at any point, if there is any issue with audio or if with the slides or if some clarification is needed, anybody can unmute yourself and discuss it with me. You may also post in the chat box. I may try and look at the chat box intermittently. But in case if I miss out on addressing something, the forum is open to discussion at any point. I'll try my level best to simplify every concept wherever possible. But at any point, if you feel that things are beyond comprehension, anyone is free to discuss at any point. You may not wait until the end for question answer session. Be frank and discuss it at any point. Okay. So, beginning with the session today for the topic impact of bioisosteres in drug discovery. So now the aim here for me today would be to give an overview of the concept first, then connect it to the part of drug discovery, which in a sense would also mean what is a drug, what is meant by drug discovery, a brief overview about it, how this concept has evolved, and eventually how it is used in the current scenario, and what is its overall impact, especially with the regulatory aspect. So what we'll do now is we'll just first check in general what is drug, we'll revise what is drug, what is the concept of drug discovery, then move to the part of bioassistor so that we have an efficient foundation. And in this short span, what I would be able to do is cover the essence of the topic. So rather than going into extensive detail, now bioisosphere in itself is a very huge topic. So at the end, I'll also give you the corresponding reference, but then there are many books available for this topic. So practically it will be difficult for us to go into great depths of it, but then definitely at the end, everyone should know the concept. So what we'll do now is we'll take stepwise, we'll develop the concept. We'll first try and identify what is a drug. So if anybody wants to answer, everyone is free to answer. But then for us, if we want to consider it in layman's term, what is a drug? What is it going to do? We all have used it right from our birth. So what is the essence of a drug? Something which is used to prevent, cure, or diagnose. So some substance which is used to modify, or it can also be used to explore. So your diagnostic, say when we are giving some radioactive material to detect cancer, that is for exploring what is there inside the body. So physiological state, something routine processes, some biochemistry processes happening inside the body. That is your physiological state. And when we say pathological state, it means some normal process has got disrupted, and that is the pathology. So something simple example, if we are not well, we have fever. So normal biochemistry is happening inside the body that has got disturbed and the body temperature has changed. So that is the pathological state. So we are giving something either to modify that state. So we take paracetamol. One crocin will cure the fever, temperature resume to normal. That is recovering or modifying. And when we say exploring, what we are doing is we are diagnosing. So we can either prevent, cure, or diagnose, and whatever substance is given for that benefit, that is the drug. So this is the simplest way of understanding what is a drug. And now we switch to the next part of it, drug discovery. So what is the process of drug discovery? Now remember, this process is a very complex process and many parameters are involved in it, many stages happen. And to give you just an idea of it, if I want one molecule to enter into the market and successfully be a drug, I have to make millions of molecules. So there is a filtering process at every stage. And finally, from millions or trillions of molecules, I may land up in one drug. So that may probability always remains. So to just understand this process very briefly, for the sake of our discussion, if we consider the process of drug discovery in the current scenario, no more serendipity. Originally, it has been serendipity or chance discovery. Now, what is done is we have various advanced tools. So, artificial intelligence taking over some routine processes. We have some data available with the help of computers, that is artificial intelligence involved. We design some compounds. So, chemical structures, we design them. We do the in silico studies. That means virtually in the computer, we try and identify whether this should be a drug or not. And then if we feel that the potential remains with that molecule, we synthesize them. So actual wet lab studies. Finally, we arrive at the part of testing. So after these processes, we test it. So supposingly, I want to devise something for corona or the COVID current situation. I will have to test it on the organism. That is the process of testing. If I want to develop anti-malarial, I will have to test it on the malaria parasite. So in this process of testing, we know whether the candidate will actually be impacting or not. So supposingly we find out that whatever molecule we made has good activity, then we proceed to clinic. That means we do the further testing on animals, we would test it on human volunteers, healthy volunteers, patients, and then if it is successful, we get it as a marketed drug. But supposingly it fails in the testing, then we'll have to redo, and that is known as a reiteration process. So drug discovery very rarely would be a straightforward stepwise approach. Many a times it is a parallel process happening where you reiterate and you redevelop, design again, test again, and then finally some molecule will come to the clinic. So now we have to keep this in mind that this is one extensive process, may require more than 10 years for one molecule to come to the market. And now with this background, we will be able to appreciate how bioisosteres will help in this process, or will help in reducing the efforts, or will help in reducing the timeline, what it will do. Okay, so what we'll do now is we'll check what are bioisosteres. So as was rightly said in the initial introduction, some molecule which is inside the biological system. So this itself has two terms, bio and isosteres. So this is with relation to biology, and isostere would mean that it is something to do with the chemical structure. So this is an amalgamation of biology and chemistry taken together, where chemical structure, how is it behaving in the biological framework. But before going on to the definition, what I would like you all to look at is one wonderful example where this approach or the value of this concept will be highlighted, and probably that will pick some interest in everybody's mind to know this concept in depth. So when I take this example, this is one classic example of aminopyrine replaced with propionazole. Now, for now, don't get bogged down with the names, with the structures. Let us not go into intricacies that way. Let us look at what this concept is. So, aminopyrine, this structure on the left hand side, this was one of the primary agents given for reducing fever. Analgesics, that means these agents are used to remove fever or to reduce the temperature. Anti-inflammatory, in layman's term, painkillers. So aminopyrine was as good as the kerosene which we use right now, the drug being paracetamol. So it was first launched in 1896, but in 1922 later, this drug was banned, or it was removed from the market because it has carcinogenic potential. Now carcinogenic, that means cancer causing potential. Now imagine you want to cure something as preliminary as fever or pain, but then you are subjecting your body to something which is causing cancer. So when we weigh the advantages and disadvantages, obviously being carcinogenic will never be accepted for something as simple as causing fever or pain. And therefore in 1922, it was banned in many countries, and unfortunately it was still continued till 1970s and eventually permanently banned, because by that time in 1951, one very simple modification was done, and uh, Roche is one of the most prominent pharma companies. Roche had developed this bioisosteric modification. So when you look at the structure, you will be surprised to see what has happened is just one nitrogen replaced by carbon. Rest entire structure is remaining as it is. And now the name is also analogous. Amino was because it was having the nitrogen, versus propionazole because it is having isopropyl. So just one nitrogen replaced by carbon removed the carcinogenicity altogether. And this molecule was developed by Roche in 1951. So by then, this propionazole entered the market, and eventually aminopyrine was out of the market. Now, if I tell you that everybody is aware of propionazole, you will be surprised. But when I tell you the brand name, everybody will recollect that you have heard of it. Propionazole was present in Saradon. And I guess everybody, even from the younger generation, must have heard of Saradon. Eventually, when kerosene was not so prevalent, Saradon was the drug for curing even simplest as headache or fever. So propionazole was that component of Saradon, apart from few other ingredients. It finally was successful in the market and replaced the carcinogenic potential of aminopyrine. The concept here was bioisosteric replacement, detoxification of something which is going to harm the body. Now here, unfortunately, what eventually happened was propionazole was not carcinogenic, but it had some cardiovascular effects. It had some hematological, so it was creating some problem to the blood. It was creating some problem to the heart and related organs. So it was eventually removed from the market and banned in many countries, but not for carcinogenicity. So that main toxicity was removed. So I guess this itself highlights how valuable this concept would be for drug discovery if it is correctly implemented. And probably now, when we are trying to see why we are studying this, this was one classic example of how it was implemented. Now, when we try to check why we are studying this picture will summarize everything in essence. So we want a drug which has potency. So the optimum potency is something which we want. Potency means the drug is able to show its action. So we want something which is having optimum potency. So we want something to be in this range. The drug which is highly potent, because on the x-axis here we have something which we don't want, some toxicity, some drawback, or something like stable nahi hai. It is not having stability, it is having issues. So whatever are the issues, On the right hand side, we will have more of the issues. So we don't want to shift on the right hand of the x-axis. So wherein now we want the potency, but we want to remove the toxicity and we want to strike the optimum balance. Now every time when a person or a chemist would make a drug, it may not be the highly potent compound. So we have to optimize. And then that final optimum solution, one of the promising strategy always remains as bioisosteres. Okay, so now what we'll do is with this background or with this stage now ready for us to understand bioisosteres better, we would look into what are bioisosteres and the formal definition, the formal components. Now again, it's fine if the definition or you are not learning of the definition, fine. It's important for you to understand the concept. So what are bioisosteres? Some substituents or some groups. Remember, bioisosteres need not be compounds in themselves. They are some components, some substituents or groups which have chemical or physical similarities. Similarities with what? Something which is already there in the drug. So we have some chemical structure with us already. Some component of the structure is having some issues or is associated with some problem. We are trying to change that with something which is chemically and physically similar. In effect, what we want, same biological property. So we may not be able to achieve identical property, and hence what is said is similar biological properties. So supposingly, I want to make painkiller molecule. I'm changing the structure, but in effect, I should still get a painkiller. It should not happen that I have changed the structure and now it is no more a painkiller. So similar biological property is what is expected in the end. Now, as a strategy, when this is implemented, what all benefits this offers? It could attenuate toxicity. We have seen the example just now. It removes the toxicity. It can modify the activity. So when we are saying modify, we are not changing. We are either improving it or increasing it many folds. So which means modifying the activity of lead. What is a lead compound? Something which is ready for the clinical trials. So that is your lead compound. So it is changing the lead compound with respect to enhancement in activity. And later altering pharmacokinetic of the lead. Now pharmacokinetic may be a new term to you all. I'll briefly explain it. When we take a drug, when we take that tablet, in simple formulation is tablet. When we take a tablet, the first thing is the tablet dissolves in the body. Then it has to enter the blood, cross many barriers, enter the blood, reach the target site. So supposingly you have pain in the leg, you have taken the tablet from stomach, the molecule has to go to the leg, occupy some receptor that is protein and show the response. So pharmacokinetic means what is the body doing to the drug. So supposingly body is breaking down the drug, it is hydrolyzing the chemical, it is changing the chemical, that is metabolism. So all these parameters, absorption, distribution, metabolism and excretion. Finally the body is throwing out the chemical. None of the chemicals would stay in your body permanently. By some way or the other, body tries and throws away all xenobiotics. That means something which is not native to the body. So pharmacokinetics is one of the important branch of drug discovery, whereby regulating the parameters will give you a good output of lead. So bioisosteres is going to, or this strategy is going to help you in all these domains together. Okay, so what we'll do is we'll quickly check how this concept has evolved, or what was the pioneering concept here, or how this term was eventually coined. So this as a strategy was known right from the early 1900, where the term was not coined, but the concept of isosterism. Now this part of isosterism is dealing with only chemical similarities, something which looks similar to the chemical of interest. That was isosterism. Probably something which you'll have also learned in chemistry, where we say structure A and structure B are isomers. Similarly, we say they are isosteres. So isosterism would be relative chemical entities. But then at this stage, the relation with the biology was not established. So James Moir had initially coined the concept of isosterism. And the word of the term isostere, that was coined by Langmuir. A decade after the initial concept was evolved, the term was coined as isostere. But later, with respect to biology, the pioneering research was by Elmer. And Elmer had identified, so all of you may be aware of antigen, antibody. Thankfully, because of COVID, now everybody has that, some words are familiar to everyone. So antigen is something foreign and body creates antibodies to get rid of this antigen. So what Elmer had observed was if we are giving artificial antibodies, this is one beautiful observation. Artificial antibodies, when administered, body is making antibodies for this antigen, antibodies which are developed. So these artificial antibodies, when developed, they are not able to identify or differentiate between benzene as a phenyl ring or thionyl. Thiophene, everybody must be aware of it. Thiophene contains sulfur and the unsaturated ring. So thiophene and phenyl are identified as the same in context to artificial antigens, which means body is not able to differentiate, or the natural system is not able to differentiate between these two as different chemicals. And that was the point where it was recognized that they may be structurally different, but biologically the system is identifying them as similar. And later on in 1950, that was the point when Friedman had termed it as bioisostere. So the concept was originated by Elmer, but then it was coined as bioisosteres, and the definition therefore developed was done by Friedman. So this is a brief perspective of or the historical background of how this concept has developed or how the terms have been coined. But in a sense, what we have to now remember is bioisosteres, something to do with chemical constituents, which inside the body, biological systems will be identifying them as same. And this is the way bioisosteres are classified. We will have something as classical versus non-classical. So we will just quickly go through this so that you will be able to efficiently identify what kind of bioisostere is there in the molecule and what you can plan in case you are planning on changes in the drug discovery. So when we say classical, this is entirely based on structural similarities. I have a compound A which has group B. Now this group B therefore will be classified based on the chemical components. So we have monovalent, divalent, tri, tetra and ring equivalents. I assume everyone must be aware of this valency of atoms or something fundamental to chemistry. So we have monovalent atoms, that means these single bonding atoms where hydrogen can act as a bioisoster to deuterium and vice versa. So if the drug has hydrogen, for some benefits I want to change it, I can use deuterium. One simplest example is CH bond versus the CD bond. Body, the way CH bond is broken inside the body is faster than the CD bond being broken. And hence in the recent times, you have deuterated compounds generated and administered as drugs. This is one of the most promising field which is developed. And if you check various patents which are applied across, you will realize that you may have the drug originally present. Now the newer patent would be having deuterated analogs. Because of this widely accepted phenomena that CD bond cannot be broken as easily as CH. So in simple terms, the body will not break down this drug same like the parent. That means it will remain in the body for the longer time and therefore show the response for a longer time. Similarly, for hydrogen, I have one more option which is fluorine. We will take this specific example and therefore this aspect I will discuss later. And you also have many analogous examples with nitrogens, oxygen, where various examples here. One of them we saw a while ago, nitrogen being replaced by carbon. Although that is not a monovalent nitrogen, it happens to be of this kind, nitrogen which is substituted here. So similarly, we have various functional groups. Depending on the valencies, we have the permutation combinations. But then these are structurally similar. So basis of structural similarity is for the classical bioisosteres. Few examples we'll be taking it later. We will now switch to non-classical. So these non-classical, upfront looking at the structure, there is no similarity or there is no identity. So earlier we said that, okay, I know that it is forming single bond, it is having similar chemical properties. But here, when we look at these examples, they will be structurally distinct altogether. The only overall perspective will be they have steric or electronic properties which align, and therefore body is identifying them as same. So one of the further bifurcations is cyclic versus the non-cyclic analogs or some exchangeable groups. So one example here is when I am substituting this benzene with the non-cyclic analog versus this has been extensively used in many drug discovery programs where carboxylic acid is changed with the tetrazole. Okay. Now, for those all are interested, you all will get many research articles as well with the heading itself stating bioisosteric replacement, because some functional group inherently have some drawbacks. One of such examples even we'll be discussing today. But then to simply place it, classical bioisosteres, we have them upright looking similar. Non-classical, they have been established over some period where properties have been judged, and their final outcome is a set of upfront not looking similar but biologically behaving similar. So this is all about the classification. Now what we'll do is something which is more interesting for us to know, but then without the background we won't be able to appreciate their applications in drug discovery. So we'll take specific examples for these classes, some general examples, and then finally what is happening in the modern situation. So one very wonderful example here, as I said earlier, we'll be taking the example for H being substituted with F. Now, where is this drug used? Now, this is something which is a native structure in the body, uracil. In your earlier, maybe 12th standard or earlier school days, you may have heard of DNA, RNAs, and their nucleotide, nucleosides. So we have adenosine, guanine. Same way we have uracil. So this uracil has a hydrogen, and what is done is this hydrogen is bioisosterically replaced with fluorine. And the reason why hydrogen and fluorine can behave in the same way in the body is they have almost very close van der Waals radii. Now, although going according to the periodic table, hydrogen is first, fluorine is not in that same row. Fluorine is the last in the next row in the periodic table. But in spite of that, because of strong forces of attraction, the shell of fluorine is brought very close to the nucleus, and then overall when we check the radius, we have almost similar radii for hydrogen versus fluorine. Now, hydrogen and fluorine definitely are not same electronically. That everyone knows and everyone will agree. But the shape of the molecule remains almost same. And therefore, it is identified as same molecule inside the body. This is the origin of classic bioisoster, where they may not have similarities with respect to chemical constituent. Fluorine is strongly electron withdrawing, hydrogen is not. So chemical properties are bound to be different, but shape-wise they are going to look same. And therefore, when this molecule is fitting into the receptor is nothing but a protein, and the proteins have some cavity. Molecules have to go and fit inside this cavity. If the shape is similar, both these molecules, because the rest of the structure is same, both these molecules can occupy the same receptor site. And that is how fluorine, this molecule is a drug known as 5-fluorouracil, abbreviated as 5-FU. So 5-FU happens to behave like uracil inside the body, gets incorporated in the DNA process, but once it is incorporated, it is not going to behave like uracil, and therefore your nucleoside synthesis will come to a standstill. And hence this drug is used as anti-cancer agent because it stops the production of DNA RNA, it terminates the cell replication, and therefore it is acting as anti-cancer agent. So anti-cancer agents are also known as anti-neoplastic agents. So it is one of the most widely accepted example of anti-neoplastic agent developed by bioisosterism. So what is the advantage here which F is offering? So F not just displaces hydrogen and occupies the same property, it also changes the metabolism of the drug, and it also changes the overall electronic properties of the parent compound. And hence again, similar to the hydrogen versus deuterium, extensively explored in the current scenario is hydrogen versus fluorine. So almost 20% of the drugs which are generated right now, or maybe this number would increase eventually, many drugs are eventually fluorinated compounds because of the changes in metabolic profile, because of the changes which the fluorine offers compared to hydrogen. And that is how, same like the hydrogen deuterium, hydrogen fluorine is also widely explored by many industries, especially with respect to the regulatory aspects. So one more example now for anti-cancer agent, yet another classic example for anti-cancer or anti-neoplastic agents. So right now, whatever is there on this left-hand side, these two are the parent structures. Now, when I'm saying parent structures, this is which you all have seen, which you all have used, folic acid. I hope everyone has used folic acid somewhere. Folic acid is one of the primary constituents for cell to develop, cell to multiply. It is an essential nutrient, consider it that way. Now, folic acid is one of the component. I'll just name the enzyme. It's fine if you all are not aware of it. Dihydrofolate reductase. This is one enzyme. Folic acid is binding to this enzyme, and then in effect the cell is able to divide, multiply, cell is able to survive. Now, I don't want the cell to survive, I want to kill the cell, because in cancer, cell multiplies very rapidly. So I want to terminate this. Now in this process, what is done, as the first agent which was developed, this was the structure which was developed, this is known as aminopterin. Okay, aminopterin was the first agent developed from folic acid, where now if you check the structure is made of three components. This is the pteridine ring. This bicyclic structure is the pteridine ring. This part of the structure is para-aminobenzoic acid, PABA. This is amino, para to it I have benzoic acid, and this component here is glutamic acid. Now, all these components have been retained as it is. In the parent drug which was developed, aminopterin, you would check that I have this pteridine ring, I have para-aminobenzoic acid, and the glutamic acid retained as it is, along with the stereochemistry. Everything retained. Just one modification is the OH here has been displaced with NH2. And therefore the name aminopterin, amino for the NH2, pterin for the pteridine ring. So on the pteridine ring, instead of this OH of folic acid, I have NH2. So now aminopterin is able to beautifully bind to folic acid and therefore prevent the cell replication. Unfortunately, what was happening is this drug is not able to differentiate between normal cell and cancerous cell, and therefore aminopterin has a lot of toxicity issues. And this THF is the substrate which is generated from folic acid. So folic acid, when it binds to DHFR, it produces tetrahydrofolate. So when you check the structures, you will realize that the saturation of ring is taking place. Rest of the structure would remain as it is. So this ring has changed. So this THF is finally the component needed for replication. Now aminopterin was developed, but it had toxicity. So as a next step, what was done was, this is the drug even used till date, methotrexate. Methotrexate because methyl has been added to this parent. Originally it is NH, aminopterin has NH, methyl has been added, giving you methotrexate. Methotrexate is one of the beautiful anti-cancer agents. It does have toxicity, but then dose modulations changes accordingly have helped, and methotrexate has been repurposed recently for rheumatoid arthritis. So very small doses used for rheumatoid arthritis. In the same way, if you now compare methotrexate and the next drug which has been developed, it is just ring modification. So N can be replaced by carbon, we have seen that. So N replaced by carbons. Now ring replacement, this is the non-classical bioisosteric replacement. And that is how these things keep evolving. You cannot stop at methotrexate only. Methotrexate is definitely good agent, but then it itself also has cytotoxicity. So it is not exclusively killing cancer cell. And definitely that is a difficult thing to achieve because finally, although cancerous, the cell is still your cell. And therefore methotrexate is not efficiently discriminating cancerous cell, but still it is way better or the toxicity is way less than aminopterin. So evolving stages are methotrexate followed by the next analog. Many analogs are still being developed eventually by various replacements. And remember again, a pointer is bioisosterism is not the only thing to be done, but definitely one of the promising things to be explored. So methotrexate research will still continue till maybe you have the best combination of toxicity versus activity. Now, one more example for non-classical bioisosterism. Now, this example, which is given on the slide here, is one of the drug discovery programs carried out by Merck. Now, what was done was Merck had to develop diuretics. Now, for simplifying this class, diuretics, we want uresis to be more. That means urine production to be increased so that sodium and some unwanted elements have to be thrown out. So you are triggering urine generation, that is diuresis. Now these drugs therefore are increasing that levels of urine generation and throwing off the unwanted things in the body. In this program, this term here is high throughput screening. HTS is high throughput screening. Now, high throughput screening, that means you have a huge library of compounds. And when I say huge, it literally means millions of compounds are tested across. So the earlier thing we saw, we logically design and then test, versus this is a parallel strategy. High throughput screening, that means I have a huge data set of compounds physically there with me. I am testing them, all of them. So we have robotics development done. Widely used are robotic arms, automation. So we are testing millions of compounds in a couple of days. So Merck had done this exercise where they have identified this molecule to show good activity. So it is known as lead-like compound. Now, what is the problem with this molecule? The problem is because of this nitro. So all drug discovery scientists don't want nitro. That is an unsaid understanding here because nitro is known to be toxic. And especially nitro with that aromatic ring, be it benzene, be it thiophene, whichever aromatic, that adds on to the toxicity. And the reason here is once this nitro enters the body, we have certain enzymes present in the body which reduce this nitro group. Now, nitro, you must be aware of it, it's a reducible group. So now when it is getting reduced, inside the body you have hydroxyl amines generated. Further, you have many nitrosamines, many reactive intermediates generated. So to summarize all the problems with nitro and especially aromatic nitro groups, they in general create many reactive species inside the body that cause cancers. So nitro groups are known to be carcinogenic. They are also known to have mutagenic potential. There is a difference between carcinogenicity and mutagenicity. Carcinogenicity, cancer causing potential. Mutagenicity, that means it has potential to cause mutations in the genetic makeup. So nitro is therefore known to be a problem maker. Now, with this concept, nitroaromatic is problem. This group had two nitroaromatic, which is then even more all the lethal combination, and hence this cannot be developed by itself. It has to be modified. And what Merck had done, it had checked various bioisosteres which are possible. So many resources were used. If time permits today, I will quickly demonstrate one online freeware which is available for predicting. Swiss Bioisosteres is the site. So many tools are available, or many scientists may use their earlier background or literature knowledge. So what was eventually done for this nitro, these were the possible bioisosteres. Now looking at them, you will obviously say that there is no similarity. And that is why they are classical bioisosteres. Upfront, they are non-classical bioisosteres. Upfront, there is no similarity. between the nitro group and these corresponding groups. But when we closely look at them, these maps which have been developed here, they are the electronic potential maps. Now again, we have sophisticated softwares to develop the electronic maps. Given the molecule, it will show you where is electron deficient region, which is electron rich region, which is the electronegative or the electropositive region. So similarly, what was observed was all these structures, they have this same electronic maps. They are not identical electronic maps, but they are very similar electronic maps across these structures, which in a sense means inside the body, these structures will be identified as same. And that is how this entire program, obviously this program is still under development, but then this nitro has been avoided. And this nitro has been eliminated to give us all these analogs. One of them again will have to be optimized because nitrile again is not considered as a good start point for drug discovery. So similar other groups will definitely evolve, but then this indicates how even the industry is using it, not just routine examples which have been explored earlier. This industries use them uh, time and again wherever relevant. Now one more good example here. See, what I have done, I have tried my level best here to cover up examples which will address various aspects. So when I summarize, eventually you will realize that what all types of examples will be covering what all features. So toxicity was one aspect. Similarly, one more aspect here is metabolism. So here, celecoxib, this is one drug which is used as a painkiller. Now, originally, these are known as non-steroidal anti-inflammatory. So, steroids are one kind of painkillers. The other drugs are the non-steroidal painkillers. So, celecoxib is the newer generation kind of agents. So, originally, I think everybody must have used combiflam, which has ibuprofen. So brufen, ibuprofen, these analogs were developed as painkillers better than the paracetamol. But unfortunately, everybody, whenever you use painkiller, must have also used antacid because they also cause acidity. They have some role in some similar enzymes are there in the body. So you're trying to remove that pain component, but triggering the acidity component, and that is the drawback. So these celecoxib or the newer agents which are developed, they are known as selective COX inhibitors. So COX2, just giving you the name of enzyme for your general interest. COX2 is the enzyme needed for removing the pain component, and COX1 is the enzyme which is causing acidity or the drawbacks related to acidity. So these celecoxib kind of agents were developed for selectively affecting COX2, which means I will have a painkiller but not have the problem of acidity. So how was this developed? The parent drug, the structure is given here, which was the first in the class to be developed. What was the drawback with this drug? This had good activity. It was not the problem with activity now. The problem was with long half-life, 221 hours. Now what is this half-life? Whenever a drug is given in the body, it is a xenobiotic, so body tries and breaks it, the body tries and removes it. So the half-life is when the drug is administered, it remains in the body, it is eliminated. So till the time it is showing action, to summarize, that is the half-life. So when I administered a drug, and that is why probably now you would be able to connect it to the component, we have multiple dosing. It is not that take one drug and then cured. You have to take the drug, maybe take it after 8 hours again. Some drugs have to be taken thrice a day. So if the half-life is less, you have to have multiple administration. If the half-life is more, just once a day is fine. Many antibiotics you must have used, you just take it once a day because the half-life is sufficient. Now imagine half-life of 221 hours. So that much time the drug is still there in the body, not removed from the body, which in effect also means along with giving the necessary action, it will go throughout the body and cause damage to the other structures as well. Not acceptable half-life, way beyond acceptable limits for the half-life. And therefore this drug had to be modified some way. One modification was this fluorine was replaced with CH3. Okay, now here at this stage, it is not a bioisosteric replacement directly, but then this was how the parent was developed. So CH3 was developed as a metabolic hotspot because fluorine cannot be metabolized easily by the body. CH3 has its own pathway. Body can break down that CH3 much faster. So once that CH3 is incorporated, I call it as metabolic hotspot because body can quickly recognize, break it down. And therefore, now check this beautiful change from 221 hours. We have brought it down to decent 3.5 hours. So which will be twice a day dosing. Fair enough. But compared to 221 and having toxicity, this is much better. And this was the stage where celecoxib was developed. Now celecoxib, apart from having the selectivity, had some issues. So metabolic changes were then done further. So if you check closely, I have one ring transformation taking place. Again, CF3 being replaced with CH3. That is bioisosteric replacement. This ring changed with the corresponding bioisostere. From celecoxib, we arrive at valdecoxib. And then the newer analog, etoricoxib. So etoricoxib is still there in market in some combination drug. Maybe few of you all may have used it. So what is done again here is etoricoxib, change the ring, and again change this benzene replaced with pyridine. Yet another example of bioisosteric replacement. So that is how across various fields, this is a promising strategy which has been widely explored and still being explored. It is not that it is an end to the story. So to convey what are the advantages or in what way bioisosteric replacements can change the properties of the drug. The first and the important property is it is definitely going to change the size. It may change the shape of the molecule, may change electronic properties. Although we say hydrogen and fluorine are similar, they are not identical. So the size, shape is going to change. Electronic distribution will change. Since the electronic distribution is changing, the amount which is getting solubilized in the lipid changes, water solubility will also change. There are many parameters across the drug which will get changed. And in effect, therefore, what we say is I have structural changes. Because I'm changing the components, obviously, we will have structural changes. The structure being changed, therefore, will also change the way this molecule is binding itself to the target. So I'm changing the receptor interactions. Since I'm changing the structure, I'm changing pharmacokinetics. That means metabolism. So CH versus CD, one of the classic examples where metabolism is changed. If I'm changing water solubility, lipid solubility, absorption also changes. So in effect, changing the structure will also change pharmacokinetics. Whether it is positive or negative, that you have to have a best judgment of it. And definitely metabolism as a subset of pharmacokinetics. Major influencer is metabolism because many drugs have activity, but they fail in the clinical trials because of the problem of metabolism. So metabolism, again, it is an extensive topic in itself, but metabolism can be beneficial in a way to the body to throw away the drugs, but then in certain cases, the drug is getting metabolized and I have toxic compound generated in the body. And that is how metabolism has to be closely studied. It's one of the primary reasons why drugs fail in the clinical trials. So all these advantages can be offered by something as a simple strategy as bioisosteric replacement. Although I'm calling it simple, but then it's not as simple as it may upfront look. It has to be a lot of literature search, lot of trial and errors, many computational tools also involved. Now, in a sense, when I have structure A and I have done some bioisosteric replacement, I'm generating a new compound in itself. So based on how novel this newer compound is, whatever you are developing, it also has implications in regulatory aspect. So I may have a patent for my molecule A. I have done bioisosteric replacement, which is completely unique, which nobody has explored yet. So whatever new compound I develop, I can have a separate patent for that molecule as well. So this is how, that is why many companies are interested in this strategy because of the implications in regulatory aspects as well. So apart from giving benefits to the drug, I can also have its value in the from the point of view of market, from the point of view of regulatory aspects. So these are the modern tools which are used, and maybe if time permits, this is what I was talking about. This Swiss bioisostore, I will quickly just demonstrate one random molecule so that those all are interested can try out themselves, because it is a freeware, anybody can explore it. So apart from that, there are certain paid databases. As said, Merck and all those companies, they rely more on the paid databases which have more authenticity, and therefore we have many more developing in silico methods as well, but these are some standard methods which are developed. And coming back to how we can now implement this in drug discovery, so I said that it will be a cyclic process of having the drug, developing it, testing it. So if we identify that the drug has some issue, I can identify the complementary structure, check the biosensor, check how it is performing. So do all the steps again, test it, but then I have validated this with the help of virtual library or the computational in silico methods. So I have a better chance of having a better drug. So I do the testing, I do the data analysis. In case I have a good output, I can put the molecule to clinic. If not, I can further explore it, because few things may be predicted by the software but may not always be applicable or may not always be accurate, and therefore I may need reiteration process as well. But in effect, what I am doing is, if I do this exercise in advance, I may have I may be spending lesser time, because I am eventually avoiding all the drawbacks in advance. In effect, therefore, I will always be saving the cost on drug discovery. Company has to spend millions of rupees, or whatever is the currency, has to spend a lot on drug discovery process, because it is from scratch and involves a lot of steps apart from manpower. And therefore, if I identify these things in advance, I may be saving on the time, I will be saving on the cost, saving a lot on efforts. So if I judiciously plan it, I could land up in one of the promising strategies for drug discovery. So, in a sense, we have tried to cover various examples which will have impact on activity, impact on metabolism, impact on physicochemical properties, biochemistry. So, for those all are interested, these are the reference books. One, there are definitely many more books, but whatever data I have presented, I have taken these from the reference articles. Two of the promising have been represented here. At any point, if you want some additional information, you are free to contact me. I will share the necessary inputs with you all. And if there is some time left, can I just demonstrate that website?
Yeah, please demonstrate, madam. You can
Okay.
go ahead and show this side.
Okay. Thank you, sir. Thank you. Okay, so whatever link was pasted, this was the site which was there on the PowerPoint, which is again a Swiss database. So it's one of the authentic databases which has been evolved. Giving a brief background about this database, earlier literature molecules have all been compiled here, and the corresponding data has been accumulated here. So what we'll do is, we'll take up one very simple example here. This is the structure generator. So you can draw structures over here. So what I am doing right now is, I am checking, I have a nitrile, taking one very simple example of nitrile. I want to replace the nitrile because I know nitrile can be toxic and I want to change it. So I am indicating an R over here, which is a general, anything could be R, aromatic ring, aliphatic ring. I am predicting I want to replace nitrile. So I will give a command to the software, find out from the database what is used instead of nitriles, and then you have all the, based on how many molecules are available, your data set will therefore be generated. Okay, I'll just share the other side. It has redirected to the next slide. Okay, is this new page visible? I have toggled between the page. So it is developing the possible outcomes for nitriles. So it will give me whatever data is available so far. Okay, it may take some time because it is a very general structure. we have this scaffold again is widely explored. There is a huge data available on what else can be used instead of nitriles. So what this software eventually will tell me is, in case I want to replace nitrile and I want to arrive at a molecule which has high lipophilicity, I want to give this molecule to brain, and therefore it has to be soluble in the brain compartment. So it should be highly lipid soluble. So when screening, if this partly it is visible on the screen, I have this delta log P. So I will check for the molecules, I will filter them, various filters are available. So I'll be able to filter them such that I arrive at only one of the set of the log P value, then check what all possible combinations are there in the site. Okay, just a minute. Meanwhile, I can alternatively also check. I think it is taking time to load. Huh? Okay, I think the screen should be visible now. So it will give us various filters. So if I want to develop nitrile analogs, but I don't want very high lipophilic, I can select accordingly based on the log P value. So it will give you the cluster of log P values, which is giving us an idea of lipophilicity. So same way, it is giving me all the possible substitutions. So it is telling me I can use ethers instead of nitriles. I can give this trifluoromethyl compounds instead of nitrile. So supposingly, I have some method to synthesize this. So I say that I have a method to make amides, I have a method to make some functional group. So I can click on that functional group. When I click on that functional group, I will get an added information about that specific functional group. So I'm trying to share the new tab which is generated where that additional functional group, whatever data is available. So when I say data is available, it will give me specific examples. So which molecule was the original molecule? What was the newer compound developed? In this newer compound, what was the activity? Has it been retained? Has it been changed? So similarly, so by the time this page is getting loaded, we'll just quickly glance through the rest of the molecules. So as simple as alcohol. So something similar to the original parent, but having alcohol would be much simpler to develop rather than having that nitrile. So I can have alcohols, sulfones, all these are the possible combinations which can be substituted for the nitrile. So that R1 is your parent, nitrile is substituted with what is given here. And now when I check specifically, supposingly I have a method to make amide available at my end. So instead of nitrile, I have to substitute. So this is giving me the example that this parent had nitrile, amide was made by the developing agents. What was the impact on activity? So supposingly I'm interested in knowing further, I can click on that and I can read about it and check what is the impact. So same way, it will give you all possible modifications. So again, this structure which I'm trying to point out, rest of the structure would remain same. Only CN displaced with CONH and what is the impact. So all these informations is all the data set, if it is made available in advance, I can predict prior to synthesizing. And therefore, that is how I can reduce the overall timeline of drug discovery. I can reduce the possibility of fallouts. The major problem, when I said at the start of the session, I may have to make millions of compound so that I may arrive at one. So that may is the bigger danger. It is not that I will definitely arrive at one drug. I may arrive, and therefore, more I can screen in advance, the better chances of fallouts. And hence now you may try and explore it for various functional groups based on your needs and prevent all the drawbacks which are there for the parent drug. Okay, so that is all at my end. At any point, if you need any additional information, you all are free to discuss with me at any point. And if there are any questions, we can always discuss them.
Thank you so much, ma'am. It was really wonderful explanation and especially those informative and motivating words. Now, I would like to request our cherished Head of Chemistry Department, Dr. Anita ma'am, to speak few words. Over to you, ma'am.
Thank you, dear. Thank you, Lakshmi. Uh, It's really a matter of interaction actually. It's the molecular signaling interaction, I would say. Excellent talk, Madam Arundhati, Madam. Um, I just give the little bit gist about your talk to the others. Uh, the talk is on the impact of bioisosteres in drug discovery, and for any drug designing or development, test is necessary on the organism to give the impact, and before processing any clinical purposes. So there are stepwise approach in the testing, like in vitro and vivo, where the human is basically targeted for this pharmacokinetic leading. So, Madam has explained very nicely the attenuation of the reaction, where the she has given the review of some compounds, and also she has given the increasing manifolds of over all the history of drugs from 1909 till today, and also she has given more efficient molecules which are safer for the treatment to the human being. And also she explained the modification which we call chemical modification, where the nitrogen atom is replaced with the carbon atom. We can say it is a receptor interaction between the molecules or active components. And she also given the nice perspective of the drugs, where she explained the classical as well non-classical bioisosteres, where also she has given the applications of the molecules. And with the when she has given the applications, where she purposely actually stress upon some active ingredients, and also given the information about the software, and the PABA molecule, which is analog with the folic acid, and how the molecule is changing its way, or you can say the logical pathway towards the drug discovery. Madam, thank you so much. It's excellent talk, and I may have some like remove some, maybe the some active components maybe, but what I recollect now, I just told here. And really, you share your knowledge with our stakeholders, and we really happy to have you as a guest to with the this colloquium lecture of Let Dr. V.N. Bedekar series. Thank you so much, and we expect your cooperation for the coming some lectures may be conducted by our chemistry department of B.N. Bandodkar College of Science. Thank you so much, ma'am.
Thank you, madam. Thank you. It has also been a pleasure to interact with you all. And definitely at any point, whatever help is needed, I'm always available. Thank you, madam. Thank you.
Over to Lakshmi.
Thank you so much, ma'am. Now I requested Honorable Principal Dr. Moses College, sir, to speak few words. Over to you, sir.
I thank you, madam. I thank you, Arundhati, madam. It was a wonderful talk, and we gained a lot out of it. Thank you so much for being with us. And do join our extended family of faculty for.
Thank you, sir. It's an honor for me, sir. Thank you, sir. It's a true honor for me to be invited that way. Thank you.
Yeah, thank you so much.
Thank you so much, sir. Oh, thank you so much, sir. It's really difficult, but the sunrise had to set. Similarly, here is the moment where we have to say goodbye. But every end has a new beginning. Let's move towards a note of thanks to all those who have really worked hard for this today's event. Before that, I request everyone not to leave the event without filling the feedback form. In the link will be provided soon in the chat box. I request my friend Sneha to propose a formal vote of thanks. Over to you.
Thank you, Lakshmi. The best and the beautiful thing in the world cannot be seen or even touched. They must be felt by heart. Thank you is one such prayer among them. I consider it a great privilege to propose a vote of thanks to all the dignitaries who have witnessed this memorable and successful event. Hello everyone, this is Sneha Tiwari from TY Chemistry Department. On behalf of Chemistry Department, I extend a really hearty vote of thanks. I would like to thank our beloved principal of BPM's BN Bandodkar College of Science, Dr. Moses Collet sir, for his presence. We will ever remain grateful to you, sir, and we thank you for being us today. Next, I would like to show my gratitude towards our most loving head of department, Dr. Anita Goswami Giri ma'am. Under her guidance, this event has achieved a successful meaning. All words of wisdom that radiated a source of energy within us. Thank you so much, ma'am. It was really great to have you with us in this event. I would like to thank the guest of event, Dr. Arundhati Abhyankar ma'am, who had wonderfully explained us about impact of bioisosteres in drug discovery. Thank you, ma'am. Heartfelt thank goes to all those teachers and second parents always stand by us, guide us, lead us, and motivate us. I feel proud and thank you for making us feel happy with your presence on this function. And last but not the least, I thank all my fellow friends for showing their interest in this program. Once again, thank you very much to assemble here. Thank you.
Thank you, Sneha. Now I request everyone to switch on their cameras so we can capture this memorable moment. Thank you, everyone. As per our culture, every event ends with our national anthem. I request you all to stand on the place for national anthem. Bharat Mata Ki
Jai, Jai.
Jai. Hereby I declare the lecture is now officially over. Thank you so much, everyone.